Good afternoon, students. Today we are going to study Lac Operon. The word lac stands for lactose. So we are going to study lactose operon of E. coli. It's a part of gene, a complete genome of E. coli. Now, lactose operon is with the help of lactose operon, we are going to study the concept of gene regulation or regulation of protein synthesis, how it takes place in bacteria. Right. Now, studies related to bacterial genetics indicate that not all genes function by determining the structure of a given enzyme. There are certain genes which regulate or control the functions of another gene. Such genes which regulate the function of other genes are called as regulatory gene. The concept of gene regulation has come from the studies of lactose operon of E. coli. The studies were carried out by Jacob and Monod in the year 1961. Okay, the studies that were done by Jacob and Monod and other investigations established the fact that in prokaryotic organisms, genes are regulated at transcriptional level. Transcriptional level means synthesis of mRNA. Okay. So to explain how regulation of gene expression takes place at transcriptional level, Jacob and Monod proposed a hypothesis which was called as Operon hypothesis. Now, what an operon is and what are the properties of an operon? Let us study that. The term operon is used to designate a group of functionally related genes. In bacteria and bacteriophages, different genes concerned with the same triad or related functions are often found clustered together in a group known as operon. Okay, so what is an operon? A group of functionally related gene concerned with the same triad clustered together is called as an operon. For example, proteins determined by the genes of an operon are usually the enzymes that catalyze sequential steps in a metabolic pathway. So let us understand this by taking example of lactose operon of E. coli. See, a bacterial operon consists of two or more distinct genes, right? Two or more genes are present in an operon. They are ordinary genes in the sense that missense mutation in one gene does not affect the other gene, but they are not like ordinary genes in the sense that all the structural genes are co-transcribed into a single mRNA called as polycistronic mRNA or polygenic mRNA, and it starts from promoter site. Okay. The proof is that the mRNA of lactose operon has a molecular weight around 1.7 into 10 raised to 6 Daltons, which is much more needed than required to encode a single enzyme. That means the mRNA, which is very long, stores information for synthesis of more than one enzyme, and hence we came to know that the operons the structural genes transcribed into a polygenic mRNA or polycistronic messenger, having several distinct translational start signals with codon AUG. At a given time, because there are multiple number of start codons and stop codons, so at the same time one ribosome will be attached to mRNA and will be involved in the synthesis of first enzyme like beta galactosidase. The second will be involved in the synthesis of other enzyme like galactoside permease. Okay, so simultaneous synthesis of enzyme will take place because there are multiple start signals. Right. The bacterial operons have certain characteristics. That means the component genes of an operon are clustered in a genetic map and different proteins specify different proteins that are concerned with the same triad. Okay. So functionally related genes are clustered together. Operon genes are independent of complementation test and are co-transcribed into a single mRNA. Nonsense mutation in promoter or proximal portion of the operon exerts polar effect on the translation of distal genes. Okay, and the phenotypic expression of a component gene of an operon is regulated by promoter operator and regulator genes. That means there are promoter genes, operator genes, regulator genes which control the expression of the structural genes. Okay. Now let us come to the main part, that is the structure of Lac Operon. The lactose operon is a part of the whole chromosome of E. coli. The lactose operon consists of, look at this diagram. This part where I have written R is the regulator gene. So the lactose operon consists of regulator gene. P stands for promoter gene. O stands for operator gene. Okay. So if you look at the structure, there is a regulator gene, there is a promoter gene, there is an operator gene, and there are three structural genes. There are three structural genes, Z, Y, A. Okay, there are three structural genes, gene Z, gene Y, gene A. Okay, so regulator gene, P for promoter gene, O for operator gene. So regulator gene, promoter gene, operator gene, and three structural genes. Okay. So the out of the three structural genes, all the three genes co-transcribed into a single mRNA. Okay. So gene Z, gene Y, and gene A. Gene Z, gene Y, and gene A, they all get co-transcribed into a single mRNA. Okay. All the three together get co-transcribed into a single mRNA. And then starts the protein synthesis. So there are multiple number of start signals and stop signals. Right. So first the gene Z synthesizes which enzyme? Beta galactosidase enzyme. Gene Y is responsible for the synthesis of galactoside permease. It is also called as Lac permease enzyme. Okay, so of the three structural genes, Z gene synthesizes beta galactosidase enzyme, Y gene synthesizes Lac permease enzyme, and A gene synthesizes trans-acetylase enzyme. Okay, so there are three different enzymes synthesized by three different structural genes, but all the three structural genes get co-transcribed into a single mRNA. The first one synthesizes beta galactosidase, the second synthesizes Lac permease, and the third synthesizes galactoside trans-acetylase. What is the function of beta galactosidase enzyme? It breaks lactose into glucose and galactose. Okay, so disaccharide is broken into monosaccharide, right? Galactoside permease or Lac permease is responsible for uptake of lactose inside the cell from outside environment. Okay, pumps lactose inside the cell. Galactoside trans-acetylase, its function is still not certain. What is the role of trans-acetylase? Trans-acetylase is not directly involved with the uptake or breakdown of function of lactose, but it is supposed to be eliminating certain toxic thiogalactosides. Okay, so its function is uncertain, that it is not directly related with lactose functioning and all, right? So this is about the structure of Lac Operon, right? Okay. So the Lac Operon of bacterium E. coli has been well studied. It has three structural genes. What are those? What are the three structural genes of Lac Operon? Gene Z, gene Y, and gene A. Z gene codes for beta galactosidase enzyme having a molecular weight of 125 kilodaltons. Beta galactosidase enzyme is made up of 4 units. It splits lactose into glucose and galactose, and the gene is 3510 base pairs in length. Y gene is responsible for synthesis of galactoside permease enzyme having weight 30 kilodaltons. It is made up of a single unit. This protein is responsible for uptake of lactose. The gene Y gene is 780 base pairs in length. A gene codes for the enzyme thiogalactoside trans-acetylase made up of 2 units. Function is not certain, but responsible for removal of toxic effect of thiogalactoside. Okay, it is 825 base pairs in length. The enzyme may not be directly involved in lactose metabolism. Okay, so just look at this diagram again, once again. So in the beginning, regulator gene. Okay, regulator gene produces mRNA, which produces a protein called as repressor protein. Okay, Then there is promoter gene, the yellow color, and okay. So regulator gene responsible for synthesis of regulator protein, also called as repressor. Right. The second is promoter gene. Promoter gene is the binding site of RNA polymerase enzyme. The next one is operator gene. Operator gene is the place where the repressor protein or regulator protein synthesized by regulator gene goes and binds. Okay. So the regulator gene produces the regulator gene produces repressor protein. It goes and binds the operator. Right. Structural genes. If the RNA polymerase is able to move from its binding site of, when the RNA polymerase enzyme binds the promoter gene, moves ahead, moves to the structural genes, and the Z gene, the Y gene, the A gene, that together transcribed into a single mRNA, and then protein synthesis starts. That is beta galactosidase enzyme, permease enzyme, and trans-acetylase enzymes are produced. Okay. Now, when we will study the regulation, we will study what is the function of repressor and lactose and all and all. Okay. Right. So, this is regarding the structure of Lac Operon, the same thing. Okay. Now, promoter gene. The promoter gene is the site where RNA polymerase enzyme binds. Okay. So, in case of a Lac Operon, the promoter gene is the site, or it represents a sequence of bases recognized by DNA dependent RNA polymerase. Rather, we can say promoter gene is the binding site of RNA polymerase enzyme. Okay, so binding of RNA polymerase enzyme to the promoter gene initiates transcription process. Okay, the promoter polymerase binding initiates transcription of neighboring structural genes. Okay, now these promoter genes is regulated by certain specific proteins known as regulatory proteins or repressors, which are synthesized by regulator gene. Okay, so the first is regulator, promoter, operator. Okay, promoter gene is the binding site of RNA polymerase enzyme, right? Promoter gene is the binding site of RNA polymerase enzyme. Okay, here, and RNA polymerase bound to promoter starts the protein synthesis, but the promoter gene is controlled by the regulator protein or repressor protein that is synthesized by regulator gene. Okay, repressor protein is synthesized by regulator gene. It controls the binding of RNA polymerase to, or it controls the function of promoter gene. Right. In the presence of repressor molecule is the protein produced by regulator gene. In the presence of, okay, look at this diagram. In the presence of repressor molecule, which is produced by regulator gene, regulator gene produces a repressor protein. The repressor protein goes and binds the operator gene. Okay. As soon as the repressor protein binds the operator gene, the RNA polymerase enzyme, which is bound to promoter, is not able to move ahead. Okay. That means the binding of repressor to operator blocks the movement of RNA polymerase enzyme. If the RNA polymerase enzyme is not able to reach the structural genes, the expression of protein synthesis or expression of structural genes will not take place. Okay. So, promoter gene is the binding site of RNA polymerase enzyme. Promoter gene, binding of RNA polymerase to promoter initiates the expression of structural genes and mRNA is synthesized, but it is controlled by repressor protein synthesized by regulator gene. Okay, if the repressor protein is bound to the operator gene, RNA polymerase cannot move ahead. If RNA polymerase cannot move ahead, expression of structural genes will not take place, right? So, we say that promoter gene is controlled by regulator gene or the repressor protein synthesized by regulator gene. There is one more regulator protein molecule called as CRP. CRP is cyclic AMP receptor protein. Cyclic AMP receptor protein binds to the regulated promoter and transcription is initiated. See, one thing is important, that repressor protein controls the promoter gene, okay, but there is one more protein called as cyclic AMP receptor protein. Cyclic AMP receptor protein, it's a complex of cyclic AMP and CRP protein. This complex binds to the promoter gene and initiates the process of transcription of structural genes. That means structural genes form mRNA and proteins in mRNA and enzymes. Okay. Promoter gene, the length of the promoter gene is 100 nucleotides and it is in continuation with operator gene. Then the promoter gene has a palindromic sequence. Okay, it has palindromic sequence means it has two-fold symmetry. There is a small region on the promoter gene which has two-fold symmetry. It is a palindromic sequence. Two-fold symmetry means if you read the sequence from left to right and right to left, it will read the same. That is called as palindromic sequence. And that palindromic sequence or the region having two-fold symmetry is the binding site of CRP protein, which is encoded by CRP gene. Okay. The next gene is operator gene. Okay. The operator gene represents a sequence of bases that interact with repressor protein. Okay. The protein is also called as regulator molecule, and operator regulator interaction will either prevent transcription or allow the transcription. Operator gene lies between promoter gene and structural genes. Okay. It influences the movement of polymerase from the attachment site to the structural genes. It is about 28 base pairs in length. Look at the diagram again. See, operator gene is the binding site. Operator gene represents binding site. This is the operator gene. It is the binding site of repressor protein. So, the regulator gene produces repressor protein. The repressor protein goes and binds to the operator and it blocks the operator. If the repressor protein is bound to the operator, it blocks the operator and hence RNA polymerase is not able to move, right? You can see here the repressor protein goes and binds the operator gene. If the repressor is bound to the operator gene, RNA polymerase cannot move ahead. If polymerase cannot reach the structural gene, then mRNA synthesis will not take place. Okay. So, repressor operator binding. Repressor operator binding controls whether protein synthesis or whether structural genes transcribe or not is controlled by binding of repressor to operator. So, operator repressor binding regulates the transcription process. Now, if the operator repressor binding is going to control whether protein synthesis takes place or not, then there should be some other molecule which can remove the repressor. Okay. So, there should be some other molecule which removes the repressor away from the operator, and that molecule which can remove the repressor away from the operator molecule is called as an inducer molecule. Okay, is called as an inducer molecule. Okay, so we have studied about promoter gene, then operator gene. Promoter gene is the binding site of RNA polymerase and cyclic AMP receptor protein. Operator gene is the binding site of regulator protein or repressor protein. Okay. When the operator gene is blocked by repressor protein, RNA polymerase cannot reach the structural genes and hence protein synthesis will not take place. Right? So we say that at that time lactose operon is off. Right. Now, every time the repressor will be bound to the operator and protein synthesis will be stopped. No. There is another molecule which we call as inducer molecule. Okay, or effector molecule. What the inducer or effector molecule does? As soon as effector or inducer molecule becomes available in the environment, it will pull the regulator away. What it will do? It will pull the regulator away, or the inducer molecule binds to the repressor protein. Inducer molecule binds to the repressor protein, and the repressor it brings about conformational change in the shape and structure of repressor protein. Okay, so the inducer molecule like lactose binds to the repressor protein, brings about conformational changes in the molecule, and now this inducer bound to the repressor protein becomes inactive. The repressor protein after binding of inducer lactose becomes inactive. It cannot bind the operator. So if it cannot bind the operator means what? RNA polymerase will move ahead, reach the structural genes, and the structural genes will be transcribed into mRNA. Okay. So the inducer molecule, what is the role of effector or inducer molecule? It pulls the regulator away. Okay. It binds the repressor protein, converts it into inactive form. When it becomes inactive, it cannot bind the operator. If it cannot bind the operator, RNA polymerase will reach the structural gene and structural genes will get transcribed into mRNA and protein synthesis will take place. All the three enzymes will be synthesized. Right? Now, what is the function of regulator gene? Regulator gene is that gene which produces a protein called as repressor protein or regulator protein. Regulator protein has specific affinity for operator. So the regulator protein goes and binds the operator. Okay, and blocks the protein synthesis. So this is about the structure of Lac Operon. Okay. So what is the function of regulator gene? The operator and promoter are under the control of another segment of DNA called as regulator gene. So regulator gene controls promoter as well as operator gene. The regulator gene produces a protein called as repressor or regulator protein. Repressor has potential to bind to the operator. Obviously, if the repressor operator binding is going to control the transcription, there should be an additional factor which will confirm whether the binding takes place or not, because every time the regulator gene will produce repressor protein, it will block the operator. So protein synthesis will never take place, but it should not happen. So whenever there is a requirement of beta galactosidase enzyme or permease enzyme or transacetylase enzyme, then the enzyme should be synthesized. So who is going to check whether the regulator protein blocks the operator or not? It is controlled by inducer molecule. Okay. So the inducer or effector molecule will control the binding of regulator to the operator. Right. So a regulator protein that binds to DNA in the absence of inducer molecule is called as repressor protein. A regulator protein that binds to DNA only in the presence of a co-repressor is called as aporepressor. Understand the 2 points. Regulator protein binds to the DNA in the absence of inducer is repressor. Active protein it is. But if the regulator protein binds to the DNA, that means to the operator region only in the presence of an effector molecule, that is a co-repressor, it is called as aporepressor. In the second case, the regulator protein is inactive. Okay. What is an inducer or an effector molecule? An inducer or effector molecule is classically a very small molecule like a sugar or an amino acid or a nucleotide which can bind to the regulator protein, change its ability, bring about conformational structural changes in the regulator protein and change its ability to interact with promoter or operator. Okay, so whenever inducer binds to the repressor or regulator protein, it will not be able to bind the operator gene. So Jacob and Monod suggested that repressor will bind to the operator and inhibit operon expression, or it will stop operon expression as long as inducer is absent, as long as inducer is not present. But as soon as inducer appears, but if inducer appears in the intracellular environment, it will bind to the regulator protein, pull the repressor or regulator protein away from operator and allow the expression of structural genes and protein synthesis will take place. Inducer binds to the repressor, destroys its capacity to recognize the sequence of bases on the operator gene by bringing about conformational change in the repressor protein. Okay, so look at this diagram. The regulator gene after transcription translation produces a repressor protein. Okay, so the regulator gene produces a repressor protein. It is active repressor. Active repressor, what happens? It goes and binds to the operator because it has affinity for operator. So it binds the operator. Because of that binding, the RNA polymerase which is bound to the promoter gene. Okay, the RNA polymerase which is bound to the promoter gene. RNA polymerase is bound to promoter gene. Because the repressor has blocked the operator gene, RNA polymerase cannot move forward. How will RNA polymerase reach the structural genes? It cannot reach the structural gene because operator gene is blocked by the repressor. And what will happen in that case? In that case, structural genes will not be transcribed. Okay. So, their structural genes, no transcription and translation of structural genes will take place. Now, look at the second case. Here what happens? Regulator genes has produced the repressor protein. Okay, regulator gene has produced the repressor protein. But the inducer molecules, is present. Okay, inducer is allolactose here. So the inducer molecule combines with the repressor, converts it into inactive form. How? Binding of inducer to repressor brings about conformational change in the repressor and it gets inactivated. Because it gets inactivated, it cannot bind the operator. It cannot bind the operator. That is why when it cannot bind the operator, then because of that, the RNA polymerase enzyme will move ahead. Okay. So RNA polymerase will reach the structural genes and structural genes will synthesize mRNA and hence protein synthesis will take place. Is that clear? Okay. So if inducer is present, it will convert the active repressor into inactive form and because of that it will not block the operator. The RNA polymerase enzyme bound to the promoter will move ahead, reach the structural genes and transcription and translation will take place. That means protein synthesis will take place. Okay, so that is about the structure of Lac Operon. So we have to understand what is promoter gene, operator gene, regulator gene and structural genes. Now, how the regulation takes place, how the control of operon is brought about, we will study in next class.